Welcome, ladies and gentlemen. So today we are going to start our discussion on DNA. And before we get into the structure of DNA or how we make more copies of DNA, let's just kind of take a little bit of a historical context on understanding how we got to the understanding of what DNA is, what it does in our body, and then ultimately, eventually, how we get to understanding what it looks like and how we make more copies. So we're going to start off with the experiment by a gentleman named Frederick Griffith back in 1928. And at this point in history, we know that some sort of material is being passed from parent to offspring, and that material is, in fact, some set of instructions that allow the body to understand how to look and how to function. And this is the reason that you have similar looking children to their parents, although they're not necessarily identical. This is why siblings look like each other, but again, not necessarily identical. So we understand the basics that there's something going on, but we don't fully understand what it is. Something else to put Griffith's experiments into some historical context is that he is working with bacteria. And at this point in 1928, we're really starting to understand this idea of germ theory and how these things like bacteria and viruses are able to cause disease. We don't fully understand at this point how or why that happens. We just know that it's possible. And that's kind of what Griffith was trying to figure out, is what makes certain bacteria pathogenic, able to cause disease, and why aren't others that same way. So he's working with these two bacterial strains of the same bacteria that causes bacterial pneumonia. And what he looks at are the smooth strain, which has a capsule. So if you remember, bacteria are prokaryotic cells. They have a cell membrane, they have a cell wall, and then some of them can have an additional layer outside of that cell wall called a capsule. And that capsule often helps bacteria adhere to certain surfaces. It's also really good at getting around mammal immune systems. So it helps provide this extra layer of protection for these bacteria. So Griffith is looking at this smooth strain bacteria that has that capsule, and as a result of that is doing a really good job at getting around the immune system of these mice that he's working with, and as a result is causing pneumonia. So when he injects a mouse with that live living S strain bacteria, mouse gets pneumonia and it dies. He's also working with a rough bacterial strain. So same bacteria, just different strain, different type, that lacks that capsule. Okay, so it doesn't have the set of instructions to make it. We understand that now based on how DNA works. But what he did know at the time is just it was missing this extra layer. He gave it to the mice. The mice, in that case, were able to recognize it, destroy it, and so essentially it was harmless. The mouse's immune system took care of the bacteria. Mouse never really got sick. Mouse lives. Now, I mentioned that this is sort of at our point of understanding germs and what they do and how we can mitigate the effects of them. And one of the things that we are starting to learn at this point is that if we boil water or we sterilize instruments in surgery with heat, we don't get infections, or at least we get a lot fewer of them. So Griffith takes this live S strain, right, which killed the mouse, and he heats it up, what we call heat killing. So he puts that bacteria basically in a petri dish or in a test tube and heats it up to high temperatures. And if you remember, bacteria are prokaryotes. Prokaryotes are still cells, which means they still have all of those macromolecules we talked about, carbohydrates, lipids, proteins, right? Proteins, you remember that? If I take a protein and I put it into really high temperatures, remember that term where we talked about it denaturing, right? It loses that three-dimensional shape and it can't do its job anymore. So at Griffith's time, they didn't necessarily understand proteins or denaturing proteins, but what they did understand is if they heated it up to a high temperature, it would basically inactivate it. It would cause it to not work. we now understand that by heating it up enough, we denature the proteins fall apart, they can't do their job, and essentially that bacteria is useless because it doesn't have the proteins that it needs in order to function. So he takes that heat-killed S strain, gives it to the mouse lives, right? That bacteria no longer has proteins to do its job, and so it's useless. Here's where it got interesting. He accidentally mixed some of that heat-killed S strain with some living R strains. Happened to give it to the mouse, and he ended up with dead mice. When he backtracked and realized what he did, it was a really puzzling conclusion, because how do I take something like that heat-killed S strain that resulted in a live mouse, accidentally mix it with some live R bacteria that also gave me a live mouse, put them together, and I end up with a dead mouse? How does that work? Well, Griffith comes to the conclusion that something in that S strain must still be viable, must still be around to give the live R strain the set of instructions to now essentially become living S, right? What he didn't know is what that material was. We now understand that it's genetic information. So one of the things that's interesting about heat-killed bacteria is that you can heat kill it up to a point of destroying the proteins, but you have to heat it up even further to destroy the DNA. And bacteria are kind of clever in the fact that they are capable of taking just random bits of DNA out of the environment and using it. Because DNA is DNA is DNA. It doesn't matter if I take DNA out of a human or out of a jellyfish or out of a bacteria. If I put it into a living cell, even if it's not the same cell as the DNA originated from, like I can take a human insulin gene, stick it into a bacteria, and that bacteria will now produce human insulin because insulin's a protein. It has the instructions. It knows what to do with it. It's a universal code. That heat-killed S strain, he heated it up enough to destroy the protein, so he inactivated it. But he didn't heat it up enough to destroy the DNA. So that heat-killed S bacteria just kind of dies off, DNA's floating around. That live R strain was now able to take that genetic information to build that capsule out of its environment and now it now became transformed into S bacteria. S bacteria kills the mouse. Pretty cool. Now, back this up a step. Griffith only knows that something is being transferred. He doesn't know what it is yet. And essentially, Griffith's experiments kind of fall by the wayside. Nobody really understands what's going on. They find it interesting. They kind of start to look at things here and there, but it's nothing conclusive. It's going to be almost 20 years later when a gentleman named Avery, along with his team, happened to rediscover Griffith's experiments with this S and R strain bacteria. And he again directly observes this transformation of our bacteria, right, harmless bacteria, into pathogenic S bacteria in a petri dish. And he can see this. He can see what it's doing in the sense of that it's killing off these organisms when they get infected, even though separately heat-killed R strains, or excuse me, living R strains and heat-killed S strains don't seem to do that. So he decides to start performing some additional tests on the material that is in existence here. And by 1944, most scientists agree that whatever this genetic material is that is being transformed from one bacteria to another, or from parent to offspring, has to be either DNA or protein. Okay, they've come to that conclusion at this point. So Avery is taking that new understanding along with Griffith's experiments, and he starts doing tests. He does a bunch of qualitative tests, and results show DNA is present but not proteins. He does a bunch of chemical analyses looking at the portions of elements that are present in these materials that are being moved from one strain to the other. They seem to resemble DNA, not protein. He does enzyme tests. If he adds enzymes that break down proteins, the transformation still happens. If he adds enzymes that destroy DNA, the transformation doesn't happen. So all of this information is seeming to support that DNA is in fact the material being transferred from one strain of bacteria to the other. And yet, there's not going to be a whole lot of consensus at Avery's time. Most people are going to think he's nuts. There's got to be something wrong with those tests. And think about it. We're talking about a material that at this point we understand exists in all living organisms. Everything from these bacteria in this petri dish to us as humans to every plant, every animal, everything on Earth has this genetic material, whatever it is, as its set of instructions. We get that at this point in history. We just don't know what the material is. And why do people think he's crazy? Well, go back and remember your basic biochemistry. When we look at DNA, DNA is a nucleic acid. The building blocks of nucleic acids are nucleotides. How many nucleotides do we have? There's 4. Adenine, thymine, cytosine, and guanine are the nitrogenous bases that make one DNA nucleotide different from another. So there's 4 bases. We talk about proteins, on the other hand. Building blocks for proteins are amino acids. How many different amino acids are there? There's 20. So if we think about this as a set of instructions, kind of like the alphabet, how many words can you build from 4 letters as opposed to 20 letters? Well, our alphabet is 26 letters long, and we can pretty much dream up any word we want. And that was the thought. There is no way that scientists believed at this point that there was any conceivable way that 4 different building blocks could be sufficient enough to explain the amount of diversity that exists in life. But it might be feasible that 20 amino acids could do it. So they think Avery's nuts. And his work sort of gets buried for a little while longer. What Griffith and Avery didn't understand at the time was how these bacterial cells worked. They didn't understand what cool things bacteria can actually do beyond just make us sick. So when we look at bacterial cells, right, they're prokaryotes. So they've got that cell membrane, cell wall, and they can have the potential for that capsule. Something interesting about their genetics is that they have a main circular chromosome in the center, but then they can also contain these secondary little rings of DNA called plasmids. And those plasmids contain their own set of DNA, which means they also contain their own set of genes or their own set of instructions. So they can contain genes that are sort of extras. They're not necessarily 100% required for the cell to function. So all of the absolutely you need to have these sets of instructions are in their main chromosome. Their plasmid gives them some extra stuff. One thing that we run into a lot of problems with is that a lot of plasmids contain genes for antibiotic resistance. What does that mean? Well, an antibiotic is a medication that you take to get rid of a bacterial infection. Well, if they have genes to break down those antibiotics, that means they survive it, and they're able to get around it. So we're running into a lot of problems where our antibiotics aren't necessarily doing their job anymore because we've used them so heavily that the vast majority of the individuals in that population have these genes to get around it, right? To destroy the antibiotic and keep reproducing. It really is natural selection and evolution at its best, and something that we can watch within a couple of weeks in a lab. In fact, the AP students do it in about 2.5 weeks. We can watch it happen naturally in human populations since we've used antibiotics as quickly as 6 months to a year, depending on the antibiotic and how overprescribed it happens to be. So here's my little public service announcement, if you will. You can't treat viruses with antibiotics. And we didn't know that initially when we first discovered things like penicillin. We were like, oh, this is going to be a wonder drug. It's going to cure everything. And so we were unintentionally prescribing it to people who didn't have bacterial infections. And as a result, quickly set up situations where we created this resistance in populations just sort of naturally occur in your body. You've got bacteria in and on you everywhere. There's some question of perhaps you have more bacteria in and on your body than you have your own cells. So by overusing those antibiotics, we were unintentionally forcing evolution and forcing this antibiotic resistance. Anyway, point here is that those plasmids containing these extra genes, like that antibiotic resistance, or like the ability to produce a capsule, can get transferred from one bacterial cell to another. So again, these guys are asexually reproducing. So one of the ways they needed to get around that inability for asexual reproduction to produce a bunch of genetic diversity was to find other ways to get genetic diversity into their population that was not a direct result of their reproduction. Plasmids were one of the ways that they did it. So those plasmids can actually get transferred from one cell to another via a process called conjugation. This is sort of the closest thing that bacterial cells have to sex, right? They create what we call a mating bridge. They make copies of that plasmid and they allow that plasmid to go over to the other cell. Now, they're not reproducing in the sense that no offspring are created, but they are increasing their genetic diversity by shifting around those plasmids and sharing them. What happened in Griffith's experiment was this transformation process. So when he killed that S strain, those free floating fragments of DNA that he didn't destroy because he didn't heat it up quite high enough, were able to be taken up then by the R strain of bacteria through this process called transformation. They can just take this genetic material out of their environment. It can be a plasmid or it can just be free-floating parts of DNA that they can incorporate into their main chromosome as well. This is what happened in that case. Free-floating S strain bacteria that contained the instructions to build a capsule got taken up by that living R strain bacteria. Now that R strain had the instructions to build the capsule and transformed into a living S strain and killed the mouse. It's pretty cool.